The overall goal of this procedure is to measure the tRNA asp methyltransferase activity of Entamoeba histolytica Dnmt2 Ehmeth enzyme. This is accomplished by first preparing an adequate tRNA asp substrate. As a second step, the methyltransferase Dnmt2 is purified as an active recombinant enzyme. Following preparation of the active enzyme, the Dnmt2 enzyme and the tRNA asp are mixed. The reaction is then initiated by adding the radiolabeled S-adenosylmethionine cofactor. The final step of the procedure is to stop the reaction and to separate labeled tRNA asp from unincorporated cofactor. Ultimately, results can be obtained that show the incorporation of the labeled methyl group into the tRNA asp substrate.
Hi, I am Helm from the Institute of Pharmacy and Biochemistry at the Johannes Gutenberg University of Mainz.
I'm Hofmann from the Helm lab.
Hi, I'm Serge Ankri from the Molecular Parasitology Lab at the Microbiology Department of the Technion, Haifa, Israel.
Hi, I am Ayala Tovy from Serge Ankri's lab.
Okay, so today I'm going to show you a procedure to measure the tRNA asp methyltransferase activities of the Entamoeba histolytica Dnmt2 homolog Ehmeth.
We use this procedure often in our lab to study the effect of environmental stresses, including glucose starvation, on the Entamoeba histolytica DNA and tRNA methylation status. So let's get started.
This part of the procedure describes an in vitro runoff transcription which can be used for the synthesis of any tRNA. For this purpose, a DNA template was designed with the complementary sequences of Homo sapiens tRNA asp and hammerhead